Alrighty guys, it is Jubilee here again, and I am back for the second part of your midterm review. So if you haven't seen the first part, I highly encourage you to go and watch that. We went over a Jeopardy review, and we covered some of the important things about the images and the questions that were on there. For the second part, hopefully you guys can see what I have here, and most of you have probably already seen it on eCampus. So what it is a bunch of images, right? And if you look at all of these images, they all have an A, a B, and a C, right? And so that's very similar to the questions that you guys have been having all semester, right? And that is because these images were taken from your midterm questions. Okay, so they are straight from your midterm questions. The only thing that I did that was kind of tricky is I didn't include the questions. So what we're going to do in this video is I'm going to be pointing out some things that you should really be paying attention to, maybe some questions you should be asking yourself when you're looking at these questions. Okay, and just so you know the format of the exam and everything, I did go over that in the first review video. But it will be very similar to what you guys are used to, okay? So it's still going to be the A, B, C, D is going to be A and B, E is going to be all of the above, okay? So they're still going to work like that in everything. The only thing that I do want you guys to be mindful of is just because you've seen this image before does not mean that the question you've got on the midterm is exactly the same as the question you've seen before, right? So hopefully many of you guys have picked this up, that Dr. Johnson loves to have the same image but make multiple versions of the same question off of it, right? And so that's kind of what we've done on the midterm, is some of them are exactly the same and then some of them are just a little bit different, okay? And then hopefully when you're looking at these images, you'll see that the kind of concepts that I'm trying to get you guys to pay attention to are a little more basic, okay, in terms of the scope of your questions. These are going to be the questions on your midterm are going to be a little bit more broad, okay. They're mainly going to be identification, function of what's being identified. There won't be too many very nitpicky questions, okay? So hopefully as you're going through these questions with me, you'll kind of understand kind of the level of difficulty of the questions are that I picked for this exam, okay? I didn't pick the super hard ones that a lot of you guys ask me questions about. They're mainly going to be the straightforward ones, and that is because your exam is going to be closed note, okay? And so I do realize that that's harder, and that's why I've made the questions on the midterm a little bit easier, okay? But yeah, so that's my advice, is just go through this with me, take notes, really try to get these images down. You will recognize a lot of them on your midterm, okay? But yeah, okay, so then we'll just go ahead and get started. Oh yeah, here's the other thing I was going to say is just be sure to read carefully, okay? Like I've said, just be sure to read carefully and don't just assume that you've seen this picture before and that you know this specific question's answer, okay? Alrighty, so we're going to go over all of these images, and then at the end I do have some extra images for you guys, and I will not be posting those on eCampus, but you're more than welcome to screenshot them off of the image, or you can just sit back and listen to me, but the main motivation behind that is because I want you guys to watch this video, okay? So that's that motivation, is you can not get those images without watching this video. I understand that means that you can just skip to the end of this video and watch that, but in my eyes that's still better than nothing, okay? So just sit back and take some notes if you want to on these images, and we'll go over some of the important things that I need you guys to know, okay? So this first image, some of the things that we're gonna kind of pick up on is that this first image, every choice is pointing to a different tissue type, right? So if you look, A is kind of pointing to that example we had of epithelium, right? So this is in the kidneys, and we haven't really covered the kidneys yet, but this is in the kidneys, and this was that example of simple squamous epithelium, right, in the kidneys. And so this is an example of epithelium. That's one of our 4 big tissue types. And just remember kind of the functions of epithelium, right, are going to be, lining body cavities. right? So epithelium is a lining, epithelium is a barrier. Just remember, that's one of the main functions of epithelium in here. B is going to be pointing to muscle, one of your big four tissue types, right? Specifically, this is cardiac muscle, and I know that because it has this intercalated disc, right? This intercalated disc, and we know that that's descriptive of specifically cardiac muscle, right? But if we think about the overall function for what muscle does as a tissue, because that's kind of what this image is depicting, is all the different tissues, that's going to be generation of contractile force, right? Generation of contractile force. And so it's mainly for movement and stuff like that as well. Okay, that's what makes us special as multicellular organisms, is we have muscles to help move us around. And then C is going to be pointing to connective tissue, right? So this is connective tissue. You've got a bunch of fibroblasts in here. You've got a bunch of adipose white fat cells in here. So that is all connective tissue as one of our four big tissue types. And then if we think about the function of connective tissue, we're going to think about it being the glue, right? The glue that kind of connects everything together, holds everything down, binds other tissues together. Okay, so that's one of the big functions I need you guys to know for connective tissue. Okay, for this one, you can see, so what stain is this? Hopefully you guys recognize this is a periodic acid shift stain, right? And so this stain is what's going to be turning our carbohydrates, right, our sugar-rich places, more magenta, right, more of that magenta purple. So if I were comparing maybe the sugar or carbohydrate levels in these three areas, so that's what these arrows are probably pointing to, is my hint, is not these individual specific cells, but these kind of areas, these kind of zones, right? And so if I were to look at A, I would say there's little to no carbohydrates in here, okay? Little to no carbohydrates. It's much less, it picks up much less of that magenta purple stain. If I were to look at B, right, we've got a little bit more of that pigment being picked up, right, of that compound being picked up. So I would consider that maybe a considerable to high density of carbohydrates in here in the stomach. And then if we were to look up here at C, right, this is very brightly colored, right? It picked up a lot, if not almost all, of the stain up here. And so I would consider this a very high density of carbohydrates. Okay, so that's kind of what you should focus on in this image. We haven't covered digestive system yet, so I don't expect you to know these cells, but we have covered the stain, right? And so that might be what this image is trying to pick up on. Okay, this one is pretty straightforward. So this is back to when we learned about intro to microscopy, right? And if you look at what all of these arrows are pointing to, so A is pointing to the brain, right? And if we think about our tissue types, the brain is a part of nervous tissue, right? It's mainly nervous tissue in here. If we look at B, this kind of yellow thing, in these body specimens, it does help you to know that they actually injected or stained or whatever the nerves in the body a bright yellow color. So this kind of yellow thing jutting out from the eye is actually the optic nerve. Okay, the optic nerve going back to the brain. So if you think about that, that's also nervous tissue. If you look here, these are kind of the spinal nerves coming out of the spinal cord. Okay, these kind of yellow things. So this would also be nervous tissue, right? So I've got three things and they're all pointing to nervous tissue. So hopefully that kind of helps you think about what this question might be asking about. Okay, and then for this one, so we've got three arrows. And so I'll kind of tell you, this one's mainly talking about epithelium. Okay, so if we look at what these arrows are pointing to, right? I'll tell you this is in the tongue or the cheek or someplace like that. I believe it's in the tongue. If we look up here and we try to identify, what kind of epithelium is this, hopefully you realize in the tongue, where you have a lot of friction, you're going to have stratified multiple layers of squamous, super flat at the top epithelial cells. Okay, stratified squamous epithelial cells. They just don't look the same as they do in your normal skin, your normal epidermis, because they're non-keratinized. Okay, non-keratinized. So yeah, this is going to be your stratified squamous non-keratinized epithelium. Okay, because this is in the tongue, and we have a lot of friction here. And then B, so B, remember epithelium does make up your glands as well. So B, this is going to be your serous glands. These are your serous glands, kind of the more dark colored ones in comparison to these. You can see a little bit down here, but this is the main depiction of that. And then C is going to be pointing to your mucus glands. Okay, your mucus glands. So these are the kind of, I think they look more green somehow. They look a little more green, a little more snot-like, and then they are a little more bubbly and fat. So that's how I associate them with mucus, is they kind of look more mucusy, more snotty than our serous glands. Okay, so yeah, that's going to be that one, just kind of the different things of epithelium. This one right here, so it looks like we're doing some matching, right? So we're kind of going to look at this and see. So A is pointing to simple squamous epithelium down here, right? One layer of flat cells. And then A right here is pointing to an endothelial cell lining a vessel, right? Lining a vessel. And so that is also simple squamous epithelium. Right? Endothelium is a subtype of epithelium. Specifically, it's the type that lines vessels. And remember, the type that we have lining our vessels are very flat and very thin, right? There's only one layer thick, and that's because we want nutrients to easily pass between these, okay? Whereas B, so this is depicting simple columnar epithelium, right? And then this is also simple columnar epithelium. Okay, because it is one layer of column-like cells. This is in your small intestines. This is one of those villi that we have. We've just done it at a cross-section. So it's like instead of cutting your finger, I know this sounds weird, instead of cutting your finger lengthwise in half, we're cutting like a chunk off, like a carrot. Okay, I know that's a weird way to think about it, but that's how we're looking at it. So we're looking at it kind of end on. And you can see those simple columnar epithelial cells there. Okay, C, so this is simple cuboidal epithelium, simple cuboidal, and then right here, what this is another endothelial cell lining a blood vessel. So you can see this is that blood vessel right here. And so this is a little flat endothelial cell. And so when we think of the cells that would be lining that, that would again be simple squamous, right? Simple squamous. Because we want these cells to be really flat so that nutrients and oxygen can diffuse between this, right? So we would not want them to be simple cuboidal. This is not a simple cuboidal type of epithelium. So these two actually do not match. Okay, so hopefully that can help you kind of think of what the question's asking, that we've got two that match and one that does not. Okay, so here we've got another one of the same. And so here you can see we're doing some more matching, right? And I'll tell you right now, these two pictures on the right here are of the same thing. Okay, so these are both within the bladder. And hopefully you remember the kinds of cells, the kinds of epithelium we have in the bladder, but that's going to be our transitional epithelium, right? Our transitional epithelium. That's that very bubbly kind of epithelium, and remember that's there to help the bladder expand without increasing the pressure within the bladder, okay? So if we do our little matching thing, this is not pseudostratified columnar epithelium, right? This is not pseudostratified columnar. Remember, pseudostratified columnar epithelium is the type of epithelium we find within the respiratory tract. So within the trachea and things like that, right? Not within the bladder. So these two do not match, okay? Simple cuboidal right here. And then right here we've got a little blood vessel, okay? This is a blood vessel. And so just like before, right, we said there wouldn't be simple cuboidal lining a little blood vessel. There's not going to be simple cuboidal lining this blood vessel, right? So this would be simple squamous epithelium, right? More specifically, endothelium lining this blood vessel. So those do not match, okay? So A did not match, B did not match, C does match, right? Because we said this is transitional epithelium in the bladder, the very kind of bubbly looking cells. Okay, so hopefully that helps you realize, the top two didn't match, C does. Okay, this one right here. So this one right here also has to do with connective tissue. Okay, and so as you can see here, we're pointing to some different cells within this connective tissue. He already told you these are nerves, right? These bundles are nerves. What he's asking you about are the cells lining the kind of extracellular space hanging out here around the nerve bundles. Okay, so if we look at this is a fibroblast, right? This is a fibroblast. It's making all of this collagen around it that you see. That's anchoring down these nerves. B is doing the same. This is also a fibroblast. More specifically, this is the perichondrium, right? C, so out here we do have fibroblasts lining around the outside of this nerve bundle. So they're all fibroblasts, okay? They're all fibroblasts, and they're all making all of this collagen out here. Okay, so this is that same picture, right, that I've showed you guys time and time again, where we talk about endochondral ossification, right, and we talk about those zones. And so, the ones that I'll still go over, the one that's not labeled, but that's going to be the zone of resting, the zone of reserve up here. That's where they're not really differentiated. They don't really know what they're doing. They're kind of spaced all throughout, right? Once they start lining themselves up, right, you see how they're lining themselves up, stacking on top of each other. This is where they're dividing in these neat little rows. That's going to be the zone of proliferation, right? Zone of proliferation. So that's what A is mainly depicting. B, here they're starting to swell up and get bigger, right? Swell up and get bigger. This is the zone of hypertrophy. Zone of hypertrophy. And then here, this kind of dividing line where it goes from cartilage to bone matrix is going to be that zone of calcification or zone of ossification, right? Where we're converting this hyaline cartilage into bone. Okay, this next one, so we're looking at a fetal finger, right? So this is a fetal finger, and this is what I've been, telling you guys about how hyaline cartilage is the blueprint to bone, right? So this is a fetal finger. They haven't been born yet, so not all of this bone has been turned into cartilage. The vast majority of it is actually cartilage right here, right? And then we've got our endochondral ossification occurring right here. Okay, so what these two are depicting, right? So A is pointing into kind of the middle of the bone. So this is part that has already been turned into bone. And it's depicting the middle of that. So that's going to be where we have our spongy bone. Okay, that's going to be where we have our spongy bone. And remember, in a fetus, this is where we do have a lot of bone marrow activity. Okay, so that's why it's not as dense as normal, but this is our spongy bone. Okay, B is pointing to this kind of dark pink stuff on the outside. And remember, that's going to be our osteoid. Right? So it's mainly osteoid, and so that's where we have our compact bone. So we have osteoid in our spongy bone, but there's a whole bunch of osteoid out here, and so that's how we know this is our compact bone on the outside of the bone. Okay, so that's where we're making our compact bone finally in this fetal finger. And then right here, this is pointing to kind of that differentiation point where we're turning this cartilage into bone. And so remember, that's where, if we ever want to make new bone, to grow this bone in length, we need to put down more cartilage, and then that cartilage needs to be turned into bone. So that's what this fetus is doing, right? To get bigger and stronger, and to make more of its hyaline bone blueprints turn into bone. So this is what we call, our growth plate in that bone, right? That's how the bone is actually growing in length. Is by chewing up this cartilage and converting it into bone. Okay, so this is the growth plate of that bone. Okay, so in this one, so this is nerve, right? We're looking at nerve here. And so what A is pointing to, this is going to be a ganglion cell neuron body, right? This is a cell body of a ganglion neuron. B is pointing to the same thing. Okay. So yeah, that's what we're looking at here, a bunch of ganglion cell bodies in here. And then C, these are a bunch of axons in here, right? Specifically, these are myelinated axons. In here, a bunch of myelinated axons. Okay, so here we're doing maybe some identification or something. And we've got some nerve going on in here, I'll tell you that right now. So A, what this is pointing to, so he tells you this is a blood vessel, and we're looking at this kind of thick cell surrounding the blood vessel, right? And so this is where it gets tricky between the difference between a fibroblast and smooth muscle, right? But I will tell you, all of this pink stuff that you see right here, you see how it looks like it's a part of the cell, right? And then you see how this nucleus is bigger, right? It's not as condensed as like this cell right here or this cell right here, right? The nucleus is bigger. It's this kind of nice round shape. So this is actually smooth muscle, okay? Because all this pink stuff around it is actually within the cell, right? Whereas over here, this fibroblast, that's the whole cell, and all the pink stuff around it is that collagen, okay? So this is actually a smooth muscle cell right here. B, this is a nerve bundle, right? So I already kind of gave that away. This is a nerve bundle. I know that because it does kind of bundle itself off in this nice way, because it likes to separate itself from the other tissues. Nerve's very exclusive, okay? C, this is doing the same thing. It's its own kind of little bundle that's bundling itself off from the other tissues. So this is also nerve. Okay, so both of these are nerves. You can see a bunch of smooth muscle over here, a bunch of connective tissue, and then there's some white fat, some adipose tissue right here as well. Okay. Alrighty, so this one, we're talking about more smooth muscle. Okay, so A, this is pointing to smooth muscle. And we'll learn more about this in the digestive system, but this is actually the muscularis interna. Okay, muscularis interna. And so that is made of smooth muscle. So it's this kind of strip, this thin strip of smooth muscle right here, muscularis interna, and that's what's going to control the movement of these villi up here in the small intestine. Okay, so the movement of these villi in here. B, what this is pointing to, so this is not smooth muscle. This is actually the bottom of another one of these villi. Okay, so this is what we call like the crypt of those villi. The bottom of those, where it kind of dips down, it's just kind of cut weird. So it's like if we cut it like that at the bottom, and then we're looking at it end on. So it's just a weird cut, and so that's why he always likes to trick you guys and think, well, maybe it's smooth muscle, but it's not. You don't have smooth muscle up here. You have it in these nice little strips. So in the muscularis interna, or muscularis mucosa, and then this is going to be the muscularis externa out here, muscularis externa. And so remember, for that, that's also smooth muscle, and that's where we've got muscle going this direction, circumferentially around the small intestine loop, and then we've got it going longitudinally, so it's going into and out of the page, so it's coming at you, and that's going to be going longitudinally down the intestinal loop. Okay, so A is smooth muscle, B is not, C is smooth muscle. Okay, this one, so we're talking about muscle, but more specifically, we're talking about the connective tissue that lines the different levels of muscle, right? And so let's see. So right here, we're saying this is endomysium. And remember, endomysium is the type of connective tissue that goes around each individual muscle cell. Each individual muscle cell. And so yes, we probably do have some endomysium around here, around these individual muscle cell bundles. Okay, remember we said paramysium is the connective tissue that goes around entire muscle bundles, so it's got multiple cells within it, not one individual cell, right? And so this right here, yes, this connective tissue is lining bundles, right? Bundles of muscle cells. Okay, and then epimysium, remember epimysium is the connective tissue that surrounds the entire muscle. Okay, the entire muscle, so the outermost part. And then this right here, what this is depicting, So this is, skeletal muscle. These are the blood vessels, the capillaries that run through skeletal muscle. Okay, so this would not be depicting the connective tissue that runs around the entire muscle. Okay, this is a capillary, right, in individual muscle cells. So that would not match. That is not epimysium. Okay, this one right here. So we're looking at white blood cells, right? This one, so A is going to be a monocyte. We've got that kind of light blue cytoplasm, kind of that horseshoe-shaped nucleus because there's a little dent in this side, so maybe more a kidney bean, but it still kind of has that dent to one side. So that is a monocyte, and you can see how it is kind of bigger for a white blood cell. Okay, and remember those are the precursors to macrophages, right? Macrophages, and those are our phagocytic cells. Okay, B. So B, this cell is mainly nucleus, right? It's all the same color. That's how I know it's the nucleus and not a granule within the cell. Not a bunch of granules. It's one big nucleus, all the same color. It's about the same size, if not maybe a little bigger than a red blood cell. This is a lymphocyte. And remember, lymphocytes create our host-specific response, our antigen-specific response. That's what's going to produce your T cells and your B cells, your antibodies and stuff like that. Okay. And then C, so this one, it's got that very neutral light pink cytoplasm, and then this kind of lobulated nucleus, right? Lobulated nucleus, multi-lobed nucleus, doing whatever. So this is going to be a neutrophil. And remember, these are important for bacterial infections. Okay, and then which of these three is a granulocyte? Hopefully you guys know that, right? So C, the neutrophil, is going to be the only granulocyte in here. So this doesn't apply to this question specifically, but I do need you guys to know which of the white blood cells are granulocytes. So remember, that's going to be the neutrophil, the eosinophil, and the basophil. Those are all of the white blood cells that have granules within them. Okay, so those are our granulocytes. Okay, this one right here. So we're going to be doing maybe some matching or something like that. So A, this is going to be a monocyte, right? This is very characteristic monocyte because it's got that horseshoe-shaped nucleus. It's very large in comparison to these red blood cells and everything like that, right? So this is going to be a monocyte, and be sure that you can match that with a description of a monocyte for what we consider normal in a monocyte. So large, horseshoe-shaped nucleus. All right. B, this is going to be an eosinophil. And the way I know that is because it's got these red granules and it's got this bilobed nucleus. Okay, so you can also call that a lobulated nucleus. More specifically, it's a bilobed nucleus, but either term will apply. It is one of our granulocytes, right? And those granules are going to be bright red in the eosinophil. Okay, so remember, this one turns into macrophages, so it's a phagocytic cell. B is more important for parasitic infections, your eosinophils. And then C, this is going to be a basophil, right? And I know it's a basophil because you see how it's grainy, right? That's different than in a lymphocyte where everything's all the same color and texture. That's because in a lymphocyte, all the blue that you see is actually the nucleus, right? And they have this very large monochromatic nucleus, meaning same color, one color nucleus. See all the graininess in this cell? That tells me that that's not due to the nucleus, it's due to the granules, okay? And remember, basophils are going to be the white blood cell that has those very large dark blue granules, right? They're so large and dark blue, I can't even really see the nucleus in here, but it does have a lobulated nucleus, okay? And this is another one of our granulocytes, okay? And these are more important for inflammation, producing histamine, as well as allergies. They play a role in allergies as well. So if I were to point to this cell and say, this is a basophil, and I know that because it has a large spherical nucleus, hopefully you would recognize that that is incorrect, right? That is not how we identify basophils. That's how we identify lymphocytes, is by their large spherical nucleus, right? We identify basophils by their large blue granules, okay? So just a hint there for you guys. Okay, so this one is talking about all the different types of capillaries, right? All the different types of capillaries that we have. So remember, the three types of capillaries that we have are continuous, fenestrated, and sinusoidal, right? And that's in order of how little they let through, right? So continuous is going to be the one that lets the least through, and that is going to be in places like your skeletal muscle. Okay, so this is a depiction of skeletal muscle, and then here all these little gaps are going to be those capillaries, and remember, those are continuous capillaries in your skeletal muscle, because we do not want blood running freely throughout here, okay, because that would mess up with their contraction and get in the way of things. B right here, so this one, this is in the liver. This is in the liver, and we'll cover liver after your midterm, but the liver filters your blood, right? So it needs a lot of blood, and it needs to get in contact with whole blood so that these cells in the liver can filter it, okay? So in your liver, you actually have sinusoidal capillaries, right? Those are the ones with big gaping holes, an incomplete basement membrane, right? The basement membrane is incomplete or lacking. There's big gaping holes in the endothelium, and that's to allow for a lot of blood through these capillaries. So B is depicting sinusoidal capillaries. What C is depicting, so this is a little glomerulus in the kidney, and remember, the kidney filters urine, right? And so when we're filtering urine out through here, we do want to let things through, right? We want to let things through, but we don't want to let whole blood through, right? It's never a good thing if you have blood in your urine, right? So we want to let things through, but not whole blood. That's why we kind of have the middle one in here in the glomerulus of your kidneys. That's going to be fenestrated capillaries, okay? Fenestrated capillaries right there. Okay, so this one, so now we're at lymphoid. Hopefully you guys recognize this from lymphoid. And so for this one, maybe we're doing some matching of the organs or something, right? So if we look at A here on the left, so we can look at the zoom out and everything and see what it looks like. But to me, this looks like we've got some lymphatic vessels out here, right? We've got some lymph vessels out here, because they look all squished and stuff. They're not perfectly round, and they're filled with that light pink lymph right there, right? And then we've got this kind of round thing with a lighter center, and it's towards the outside, right? So possibly in the cortex, right? And which one of those lymphatic organs has follicles or things that look like this in the cortex? That's going to be the lymph node, right? So this is a lymph node, and you can see we've got this follicle in the cortex with a germinal center. We've got this parafollicular area with those hot pink high endothelial venules, right? So this is a lymph node. Okay, and then if you look right here, this picture is also depicting a lymph node, right? This is just kind of the drawing version, or the cartoon version, right? We've got this kind of kidney bean thing, and we've got all these afferent lymphatic vessels coming in from all directions. We've got one efferent lymphatic vessel coming out the middle. We've got a big medulla and a cortex, and in the cortex we have those follicles, right? Those follicles in the cortex. So those are both lymph nodes. This one right here, so if I look at this one right here, it looks lobulated, right? Does that look lobulated to you guys? We've got all this connective tissue separating all these different chunks, and in each one of those chunks, it's got this kind of darker staining outer part, lighter staining inner part, right? So it's got a cortex and medulla. It looks like it has multiple cortexes and medullas because it is lobulated, right? And remember, the one organ that we know is always lobulated is going to be the thymus. Okay, so this is the thymus. And if you look here, this is a zoom in of the thymus, but you can also see the kind of zoom out of the thymus. But yeah, this is also the thymus. I guess I kind of gave this away. So we've got a bunch of thymocytes, right? Or potential T lymphocytes in here. And the kind of big giveaways, we've got these endothelial reticular cells, right? And remember, that's what makes the blood-thymus barrier. those endothelial reticular cells. And then here, so what this one is depicting, so this one, we've got a bunch of strands in here. You see these kind of dark pink looking strands with gaps and stuff like that, right? So this is characteristic of the spleen, how we've got this kind of strand-like appearance. Because remember, we had those Billroth strands with literal cells lining up on it. And then we've got these sinuses in here that the blood kind of percolates and filters through. Okay, so this is the spleen, and then this here is also the spleen, right? Because we've got a bunch of red pulp, we've got some white pulp, and the reason I know that is because it's not all concentrated in the cortex or anything like that, right? The white pulp is evenly distributed throughout. So that's how I know it's not like a follicle like it is in the lymph node where they're only on the outside. So this is white pulp in a spleen. And remember, the white pulp is where you have a high concentration of white blood cells. The red pulp is where you have a high concentration of red blood cells. Okay, and so this picture is talking about lymph nodes. Okay, so it's trying to describe the kind of the purpose of a lymph node, right? And so remember, we said the lymph node is a secondary lymphatic organ, right? It's a secondary lymphatic organ, meaning it is antigen dependent, meaning it needs the antigens and the white blood cells in order for it to perform its function, right? And its whole function is to filter lymph. Okay, so what happens is it gets lymph from all of its afferent lymphatic vessels. It gets white blood cells from its afferent lymphatic vessels. It also gets those from the heart and everything from the blood in your vessels as well. And the whole job of putting all of this blood and all of these white blood cells and concentrating it in the lymph node is because you're hoping you're also going to have a bunch of antigens in the lymph. So we're taking the lymphocytes and we're taking the antigens and we're putting them together in this highly concentrated place so that they can react with each other, right? So that the lymphocytes can recognize the antigen and they can start creating their immune response to try to fight off this antigen. Okay, once those lymphocytes find that antigen, right? So if there's this specific lymphocyte and it finds its specific antigen, right? It finds its guy that it is meant to do battle with for the rest of its life. What it's going to do is it's going to start dividing, okay? So what it's going to do is it's going to undergo mitosis, right? That's the normal division of a cell. So it's going to undergo mitosis, make a whole bunch more of itself so that there's way more of that specific lymphocyte targeting that specific antigen so we can really create an immune response, right? And so that's what they do when they come in contact with their specific antigen. Okay, and if they don't come in contact with their specific antigen in this lymph node, right, their guy that they're meant to do battle with for their whole life doesn't come in here, and they've been hanging out in here a while and they don't see that guy, what they'll do is they'll leave the lymph node. And then they'll get recycled and they'll end up in another lymph node. Okay, so it constantly recycles T and B lymphocytes through the lymph nodes. Okay, so these lymphocytes are constantly being recycled. They hop around from lymph node to lymph node and they are looking for their antigen, for their specific dude that they are meant to battle with. Okay, so that's what that one's talking about. And this one, so we've got a bunch of nerve. Hopefully you realize that when you look at this, right? So you've got a bunch of nerve. So this is depicting a bunch of axons, right? A bunch of axons. B is depicting a Meissner's corpuscle, and remember we said that's a receptor of nerve. That's your light touch receptor. And then C, we've got a bunch of neuron cell bodies, a bunch of ganglions, right, in here. So if we think about this, we've got a receptor, an axon, and a cell body in this question, right? And so that's kind of all the three big points when we think about the functions of the nervous system, right? So if we think about the function of the axons of the neuron cell, remember that's mainly transmission, right? Axons are mainly responsible for transmission of that signal from one place to another, right? They're just gonna send it back and forth wherever it needs to go. B is important for the reception of that signal, right? It's a receptor. It receives that signal from the outside environment or wherever it needs to receive it from. Okay. C is the integration of that signal, right? The integration of that signal, because it's getting a bunch of signals from all these different receptors, and it needs to decide what it's going to do with that signal, okay? If it's going to send it off and keep the signal going, or if it decides that that's not an important stimulus to transmit to the brain. So integration, that's where it kind of decides what to integrate and what to actually send through. Okay, so this one right here, so this one is talking about kind of connective tissue. Okay, so this is an elastic artery, and remember we said, so this is specifically in the tunica media of that elastic artery, and remember we said in the elastic artery, in order for it to bound out, right, to expand in response to an increased pressure, and in order for it to recoil and contract back down in response to a decreased pressure, right, so it's this kind of pulsile movement where it gets bigger and smaller and bigger and smaller. The reason it can do that is because it's very elastic, right? It's very elastic. So just like a rubber band, if you stretch it, it can still bounce back down, right? It's easy to stretch and it's easy to bounce back down. And so the reason it's very good at that is because it has these kind of elastic things that we call an elastic lamina. Okay, an elastic lamina. So that's these kind of wavy-like proteins that allow for this elastic artery to be stretched and allow for it to recoil back down very easily. Okay, right here, this is the same thing. So this is just an EM of an elastic lamina. Okay, here it even says internal elastic membrane. That's just another name for it. Elastic lamina literally means elastic layer. So this is another elastic layer in here, and that's lining an arteriole. It's just much smaller. Okay, internal elastic lamina. And then here, this is a muscular artery, and so in muscular arteries, we don't really have this whole elastic lamina all throughout the tunica media. We mainly have smooth muscle, right? Smooth muscle. We still have smooth muscle here, but we have a lot more in a muscular artery, right, than in an elastic artery. The thing is, though, on the inside in the tunica intima, we still have an elastic lamina, okay? And so that's why this kind of endothelium, the internal elastic membrane, is kind of recoiling down because it's not being stretched from a pulse yet, okay? So we do still have an elastic lamina in here, okay? Or internal elastic membrane. It's all a different name for the same thing. Okay, so for this one right here, we're talking about connective tissue, okay? And so I will tell you right now, A is not pointing to connective tissue, right? This is the epidermis. This is the epidermis. This is epithelium, right? That stratified squamous epithelium that lines body cavities, that lines barriers, and stuff like that, right? So this is epithelium. But when we talk about B and C, these are connective tissue. Okay, and so remember B, this is in the dermis of your skin. This is where we have that dense, irregular connective tissue. This is where we have all those blood vessels and everything going on, right? So this is a site for metabolite exchange, right? Because we have all these blood vessels supplying blood to the tissue around it, supplying blood to this epithelium out here. So there is a bunch of metabolite exchange in this section of connective tissue. There's a bunch of mechanical support, right? Because it's dense, irregular connective tissue. It's really anchoring things down and gluing things down, and it's got collagen going in every which direction. And because we've got a bunch of blood vessels as well, it's also the site of inflammation, right? Because we've got a bunch of blood vessels. If a cut happens and we get a bunch of bacteria in here, remember margination is going to occur of those white blood cells. And so those white blood cells are going to line up and they're going to go out into the connective tissue and start fighting off whatever infection's going out here, okay? So all of that happens right here. C, so this is that adipose tissue, right? That chicken wire, that white fat that we have here. So this is mainly energy storage right here. Okay, so this one right here, we've got more connective tissue. Now we're just talking about individual cells of the connective tissue. And so here, so what A is pointing to, so it even tells you right here, adipocyte or fat cell, right? So adipose tissue, white fat, and then right here, this is that chicken wire, right? This is that chicken wire we were talking about with those fat cells. So this is also adipose tissue. So both of those are adipose tissue. Right here, B is pointing to a free macrophage, right? So a macrophage. And right here, we're seeing if these match, and they do not. So this right here is a plasma cell. Okay, this is a plasma cell. I know it because it has that perfectly circular nucleus, right? It's so perfect and circular. It's got a bunch of cytoplasm all around it. It looks like a cracked egg, right? With that perfect yolk in the middle. So this is a plasma cell. And remember, what plasma cells do is they produce antibodies, right? These produce antibodies. Remember, what macrophages do is they're in charge of phagocytosis. So these two do not match, okay? They do not match in identification or in function. See, this is pointing to a fibroblast, and then this right here, this is an EM of a fibroblast, okay? It's got that very condensed nucleus, and it's got very little cytoplasm around it. Okay, you can't really see much cytoplasm around it. So this is a fibroblast as well. And remember, what those do is they are the collagen producing cells of connective tissue. Okay, this one right here. So we're talking about nerve. And so what A is pointing to in the diagram and in the picture is this kind of cell lining the outside of this nerve axon. So this is a nerve axon, and this is the cell lining the outside of it. And so that's what's happening here as well. So this is a fibroblast, okay? This is a fibroblast lining an individual axon, and so that is considered endoneurium. So it's making all of this connective tissue, all of this collagen around this axon. So this is a fibroblast. Also the endoneurium, making the endoneurium. And then B, so what this one's pointing to is it looks similar, and I know it looks similar, and that's what's tricky, but see how this one is hugging right around and connected to the myelin around this axon. So this is the axon, and see how this one is specifically like hugging it, unlike this one, right? Unlike this one, right? Unlike this one. So yeah, so this is a Schwann cell. These are both Schwann cells. And remember, Schwann cells are what are going to make the myelin around the axon. So this is the axon, this is the myelin, that's that protective sheet around it. So yeah, that's what that is. And then C, so this is pointing to the same thing right here. So hopefully this is also a Schwann cell, and it is. So you see how this is kind of connected to the myelin around this axon. It's kind of making like a little hat on the axon. It's sitting right on top of it, different than this one, right? This one. So this is also a Schwann cell. Okay. Alrighty, and then this next one, so we've got this kind of diagram, right, depicting the different pressures and surface area of the different vessels within your body, right? And so this one right here, the lowest surface area and the greatest difference in pressure, right? We've got a pretty bounding pressure here where it goes from 120 to 80. What A is depicting, remember, that's going to be your aorta, okay? Your aorta, the largest artery that you have, the aorta. And then we're going to go down, and as the pressure decreases, that's where we go away from your aorta and we start to have your large arteries, okay? Your arteries. So right here, so that's where the pressure is down a little bit. The surface area is starting to increase, right, because you have more than one of them instead of just one aorta. Right here, this one, I know there's no letter choices here, but I'll still tell you. Right here, where it starts to focus down and the difference in pressure starts to decrease to where there's not really a pulse anymore at the beginning, right? Because the pulse is the 120, 80, 120, 80, 120, 80. Where we start to have a lot of resistance to that pulse, right? That peripheral resistance to that pulse, that's where we're going to have our arterioles, right? And that's something really important you need to know about arterioles, is they provide that peripheral resistance to that pulse, okay? B right here, so this is where we have the largest surface area, and we have absolutely no pulse, okay? That's going to be where you have your capillaries, okay, your capillaries. And remember, we want the largest surface area because this is where we're actually exchanging oxygen, nutrients, stuff like that. So we want a large surface area to be able to exchange all of that. So that's why your capillaries are right here. Right here, this is going to be your veins, okay? Your veins, your small to medium veins. So this is where the surface area is starting to go back down, the pressure is going down, and then once it gets kind of negative, that's where it's like your very large veins, so your vena cava, okay? C is going to be more your vena cava. And it does need to be negative so that can go back to the heart, because remember, the heart during rest is at a pressure of 0, and then it creates a pressure of 120, okay? Alrighty, so that is the end of the ones that I have posted for you guys. I will go ahead, as promised, and go over some kind of extra ones. Just because I can't help but give you guys more help. I don't know, I just want you guys to do well on your midterm, okay? So study really hard, study these images. You'll see once you take your midterm how nice I'm being, because I did not used to do all of this. I used to just do the midterm review, and I just kept getting nicer and nicer, and so now I'm giving you guys even more images. So here's a couple more. There's not too many more. Let's see, there's like 5 or 4. So we'll just go over these really quickly as well. And again, I'm not going to post these on eCampus. Feel free to take a screenshot of the screen if that's what you want to do to study these. I don't really care. My reason for doing that is because I want you guys to watch this whole video. I do want you guys to pay attention. I want you guys to learn, obviously. So that is why it's not posted on eCampus. Also just because it's kind of an extra anyways. It's already me being nice, so don't say I'm being mean by not putting it on eCampus for you, okay? Okay. Get down. Okay, sorry, my cat's driving me crazy. So A, what this is pointing to is kind of this outermost lining around this whole entire cell. Okay, so this is one muscle cell. Okay, this is one muscle cell, so this is that outermost lining. So this is the cell membrane of a muscle cell. And so kind of the special thing about a cell membrane of striated muscle cells is the cell membrane will actually come and dip down into the cell. Okay, it will dip down into the cell, and that's what forms that T-tubule that we've been talking about. Okay, that's what forms that T-tubule. So B, what this is pointing to, this is a bunch of T-tubules. All of these are examples of T-tubules. That's that part of the cell membrane that dips down into the cell and allows for the action potential that's going to be on the cell, that exciting electricity kind of thing that tells the cell to contract. The T-tubule allows that action potential to penetrate deep into the cell. Okay, so we have that action potential all the way throughout the cell. So every single part of the cell is being told to contract at the same time. Okay, and then surrounding those T-tubules are going to be what C is pointing to, the sarcoplasmic reticulum. The sarcoplasmic reticulum. And remember, we said that's kind of the site of storage for calcium. And whenever the sarcoplasmic reticulum gets that action potential from the cell membrane, from the T-tubule, it's going to be told to release calcium. Okay, so that is what the sarcoplasmic reticulum does, is it holds and stores the calcium and then releases the calcium when it's told to help regulate muscle contraction. Okay, this one right here, so we've got more white blood cells. Now we've got white blood cells in an EM and in light microscopy. So we can see here, and I know the resolution isn't the greatest. Hopefully it'll be better in your quiz. I was just taking pictures of the test. But here we're doing some matching between the EMs and the light microscopy version of each cell. Okay, and so remember I told you guys, feel free to go back and watch my TA review video where I went over how to do all these EMs, because I do know they're difficult, but I do want you guys to be able to do them. Okay, you will see it on your midterm, I promise. So what this one is, in the EM, or here we'll do the light microscopy first because that's easier. So in the light microscopy, we've got, you don't even need the great resolution to tell what this is, right? We've got these bright red granules, right? Even more bright red than the red blood cells around them. So this is an eosinophil, right? This is an eosinophil. And remember, an EM of an eosinophil is going to have a crystalline core granule, right? Crystalline core granules. This one right here, so it doesn't really look like those crystalline core granules we were thinking about. They're more very large granules, right? They're very large, dark granules. They're all kind of the same color within the granule, right? There's no kind of difference in texture within the granule. And it does take up most of the cytoplasm. So this is going to be a basophil. Okay, this is a basophil on EM, because remember they have those very large dark granules that are all about this, within each granule it's all about the same color and texture, and they do take up most of the cytoplasm. So that's a basophil. So these do not match. This one right here on light microscopy, so we've got this kind of light bluish purplish cytoplasm, right? And then we've got this kind of divot into one side of the nucleus, right? So that's the kind of horseshoe shaped kidney bean nucleus. So this is a monocyte. Okay, this is a monocyte. And then right here in this EM, remember, don't look in the cytoplasm and try to tell me what's going on. Look at the cell membrane, and you can see these very prominent arm-like projections, right? These very prominent arm-like projections coming off of the cell membrane of this cell. And remember, that is very characteristic of a monocyte. Because they're important for phagocytosis, they use those arm-like projections to help them grab onto antigens, grab onto cellular debris, so that they can engulf them, so that they can phagocytize them. Okay, so those two do match. This one right here, so we'll look at the light microscopy. So we've got this very neutral, light pinky cytoplasm, and then we've got this kind of crazy looking lobulated nucleus. Right, lobulated nucleus. So this is a neutrophil, right? This is a neutrophil. They've got the lobulated, multi-lobed nucleus, light neutral cytoplasm. And then this one right here, this is kind of the tricky one, but kind of by process of elimination, this is also a neutrophil. Okay, so it doesn't have those giant, large granules of a basophil. It doesn't have crazy arm-like projections like a monocyte. It does not have crystalline core, big granules like an eosinophil. Okay, and then it is not very perfectly round and cute, and it doesn't have a big nucleus that takes up most of the cell like a lymphocyte would. So this by default is a neutrophil. Okay, so this one right here, I'll tell you right now. So what this is pointing to, right, this is that break between the myelin of that one Schwann cell is making and the myelin that another Schwann cell is making. So this kind of break in the myelin of that protective coat is called the node of Ranvier. Node of Ranvier. Okay. B, that's what that's pointing to as well. So this is an axon. This is an axon, and this is kind of the myelin sheath around it. This is just in an EM. And so this kind of break or divot in the myelin coat of the Schwann cell over here and the Schwann cell over here, that's another node of Ranvier. Okay, this right here, what C is pointing to, right, we've got a little divot in the myelin surrounding the axon. So again, that's a node of Ranvier. Okay, this one right here. So I told you guys, I don't need you to know too much about the details of a sarcomere or anything like that. I really just need you to know A band is dark, I band is light, right? A band's dark, I band's light. This is kind of talking about how we, divide muscle, because remember, a muscle is a bundle of bundles, right? There's a ton of bundles in muscle, so it just keeps going down and down and down until we get to the very smallest level. And so the way that works is first you have an entire muscle. That muscle is made up of fascicles, right? Hopefully you remember that. That muscle is made up of fascicles. Each fascicle, each bundle within that muscle is made up of multiple muscle cells. So that's what this one is depicting. This is a single muscle cell, and you can tell because it's got these nuclei on the periphery, right? These nuclei on the periphery. You've got these A and I bands going throughout here. So this is one muscle cell. And remember, within each muscle cell, you've got more bundles. These are called myofibrils. Each one of these is myofibrils. So multiple myofibrils make a muscle cell. Okay? So that's what that is. And then if we break down each myofibril into the little sarcomere and we look at the little filaments that make up each sarcomere, these are going to be called myofilaments. Myofilaments. Okay, so it goes myofilaments, myofibril, muscle cell. Muscle cells make fascicles. Fascicles make entire muscles. Okay, so that's what that one's talking about. This one, this is our last image. So this one is going to be lymphatic tissue, right? So this is the lymphoid system. And maybe we're going to be identifying these little organs in here. Okay, so what A is going to be pointing to is the tonsils. Okay, so remember this is the tonsils, and remember the tonsils have a bunch of crypts. So these are all tonsillar crypts that this is depicting, right? You can really see that up there, tonsillar crypts within here. So that's what's going on in this image, right? And then we've got these little high concentrations of lymphoid tissue. Okay, so the tonsil isn't one of those big lymphoid organs we talked about. It's just another one of those organs that does have concentrations of lymphoid tissue within it. Okay, so that's what this one's depicting. This one right here, so this is that same picture we keep seeing, right? So we've got these afferent lymphatics, lymphatic vessels on the outside, right? We've got the capsule, and then out here towards the outside, we've got this kind of thing, right? This kind of round looking thing. So this is a follicle, right? And I know it's a follicle because it's towards the outside, it's in the cortex, right? It's got this germinal center, and that's very characteristic of a lymph node, right? They've got these follicles in their cortex towards the very outside of the organ with these germinal centers, right? And then we've got these hot pink high endothelial venules in this parafollicular area, right? The area around the follicle. So this is a lymph node, okay? And remember, that's a secondary lymphatic organ. See right here, so we've got, it looks like, there's this big division of connective tissue right here. We've got this darker staining part out here, this lighter staining part out here, so maybe a cortex and a medulla. And then if you look here, this is kind of the big hint. So remember what these are. These are Hassel's corpuscles. These are Hassel's corpuscles all throughout here. And then if you look at these lighter pink staining things, those are those epithelial reticular cells. Okay, epithelial reticular cells. Remember, those are what are responsible for making the blood-thymus barrier. We don't really know what Hassel's corpuscles do, but these are all very characteristic things of the thymus. Because remember, it's lobulated. It's got, a cortex and a medulla within each lobe, or at least it looks like it. So this is very characteristic of the thymus. Okay, and remember, the thymus is a primary lymphatic organ. It is responsible for making your T cells, or at least developing your T cells. But yeah, so that is the last image that I have for you guys. Some quick advice that I would give you guys when you're studying is hopefully, you saw based on the pictures that I've shown you, kind of the depth of knowledge I expect you guys to have on the midterm, how it's a little bit different, a little bit less specific than your quizzes, right? I do really want you guys to look at the questions because they may be just a little bit different than a question that you're used to, okay? Some of them will be entirely new, but those ones will be easy, I promise. And then, yeah, so that's kind of my main big hint is just, read very carefully. Some other resources, if you're studying tonight or the rest of this week, is to really pay attention, if you have time, rewatch my TA reviews, okay? Because my TA reviews are more broad, right, than the lecture videos. They are more broad. They do cover more of the big concepts, the general overall function, and the important things to identify, right? So that is kind of what I told you you'd be tested on in your midterm, are the big, general, overall, broad concepts. And so that would be a good thing to study, okay, are my midterm reviews. But yeah, other than that, don't stress out, you guys, over this test. This is why we give you so much extra credit. Oh, and here's another thing, and I'll put it in the group me. Is there is an extra credit opportunity for you guys, another extra credit opportunity for you guys for watching this review. And it will be worth 5 points of extra credit. So normally I give you guys 5 points of extra credit for watching my TA reviews. This week I will give you 10 because I know it is 2 hours long, and just because I'm very nice. And so all I need you guys to do is I will make a spot on eCampus for you to submit it. But what I want you to do is either submit your notes that you've taken during these midterm reviews, any notes that you've taken while watching this, or I want you to tell me the 3 most important or helpful things that you learned while watching these TA reviews, okay? So if you submit that on eCampus, I will give you 10 points of extra credit, okay? So yeah, that was my point, is we do give you guys a lot of extra credit, and that's why I don't want you to stress out too much about this midterm. All I want you guys to do is learn, right? So I do want to motivate you to learn. That's why, we do challenge you. We want you to take the midterm. We want it to be closed note, but I don't want you to stress out so much. So that's also why we give you extra credit. That's why I allow all of those extra points in the midterm so you can get above a 100. That's why I make this TA review, so that as long as you learn something in this TA review, you should do fine on your midterm, okay? So other than that, I wish you guys nothing but the best of luck. Good luck studying. Make sure to get some sleep. Sleep is important. And yeah, good luck on your midterm.